Well, look, it's a real pleasure to come today and talk to you guys a little bit about our a little bit about our research. We've met Tiago and James, and looking forward to starting a dialogue about more interesting projects. So I'm Albert Kim. Chris Bridge is also here. We're part of the QTIM group at Massachusetts General Hospital. And in short, the goal of our group is to explore clinically impactful questions in cancer with deep learning. Um, I have a background in clinical oncology as well as some background in genomics. And in my experience, I've seen there's limited crosstalk between the clinical oncologists, data scientists, wet lab biologists. And so that kind of leads to, each party having like an incomplete understanding of, what the other can do. And so clearly I'm biased, but I'd like to think that our group is unique because what we do is we collaborate with a number of people around the hospital to identify an unmet clinical need, um, after which we accrue a data set. This is one of the benefits of working in a hospital system. And then, explore ways in which machine learning can be used to make new biological insights. And so our hope is that these collaborations between physicians, data scientists, and biologists can lead to something that we hope is greater than the sum of its parts. um, In my opinion, I think deep learning in oncology has growing interest. I did this PubMed search where over the past few years, there's been a sharp uptick in deep learning and multimodal fusion learning. But the interesting thing is that when you think of cancer, I think a lot of people think of, functional models, mouse experiments, genomics. And deep learning is far less explored than a lot of the wet lab techniques, suggesting that there's a lot of promise in using deep learning to improve clinical paradigms in oncology. I give talks to a lot of clinical folks, and so this may not be you probably already know this. Many in the audience may already know this. So maybe about 8 or so years ago, the hype behind potential applications of deep learning as applied to medicine was enormous. Maybe I'll read one of these quotes. So, Geoffrey Hinton, who's a British computer scientist, said, "I think if you work as a radiologist, you're like Wile E. Coyote in the cartoon. You're already over the edge of the cliff, but haven't yet looked down. There's no ground underneath. People should stop training radiologists now." Keep in mind, this was said 8 years ago. It's just completely obvious that in 5 years, deep learning is going to do better than radiologists. It might be 10 years. What I think is maybe a more realistic goal is, there's still, in my opinion, a lot of hope, but the hype has cooled off a little bit. And I think this WHO news release summarizes the mood, quite well. And, in my opinion, I think the narrative of bringing AI to medicine is really just beginning. There's a lot of promise, as you saw in the last slide, but not a lot of data and proof. And this is important because, the risk of, a faulty algorithm, especially one that's a black box, dictating human health can be enormous. So this is an analogy here that I thought might be applicable to patient care. So this is essentially autonomous self-driving cars. And so there are like 5 different levels here. Level 5 is essentially full control of the car under all conditions. And level 0 is kind of what we're doing now. I think that we've seen that for cars, given certain treacherous road conditions, level 5 is probably never going to happen. And I would argue that's probably true for human health as well. And so a more likely outcome is probably here in level 3, in which we can synergize decision makings between humans and models. And so I think this tweet up here summarizes where I think the field of medicine is going. Will AI replace radiologists? The answer is no, but radiologists who use AI will replace radiologists who don't. And so we maybe with that we can start with talking about a little bit of the projects in the lab. So in short, we have a number of we've trained a number of segmentation and classification models. And again, the underlying basis for many of these efforts in the lab is to really design models that help augment clinical decision making. And so one avenue is through segmentation of brain tumors. So for an introduction, the tumor that we applied the segmentation model was for meningiomas. So here's a meningioma here. This is a brain MRI where I hope you guys can see my mouth. This is the nose up here and the eyes, and this is the back of the head, and the ears are over here. And so here we see pretty large contrast-enhancing lesion in the back of the brain. And here's another meningioma here on the right lateral side of the brain, right temporal side of the brain. And so meningiomas are actually the most common tumor in the central nervous system, prevalence of about 170,000 per year in the United States. The interesting thing is that meningiomas aren't actually a brain tumor. They're affecting the covering around the brain, which is called the meninges or the dura. And most of the time, I would say about 75 to 80% of the time, these are benign entities. But high-grade meningiomas, which comprise like the other 20 to 25% of the time, possess an aggressive phenotype and carry a poor prognosis. And so for that cohort, trials are evaluating new drug systemic therapies are being planned. But the way in which they call response is flawed and prone to high rates of inter-rater variability. And so this is the way, the gold standard way of dictating response as demonstrated by the response assessment in neuro-oncology. I'll say this as RANO from now on. So essentially what we do here, this is a different type of brain tumor, but this is applied for all central nervous system tumors for intents and purposes. So this is a axial slice of a brain MRI with a tumor here. We can see that the tumor is heterogeneously enhancing. So we can see over here, there's a lot of contrast enhancement, and here there's a necrotic interior. And so what the directions say is find the axial slice with the largest tumor area. So the radiologists do that visually. And then after this, the guidelines state to find the largest measurable, at least over 10 millimeters diameter, excluding necrosis and blood. So as you can see, I did this here, like being careful to exclude the necrotic area. And so, depending on how you called the necrosis and blood, the tumor may or may not be measurable, even though it's very clinically apparent to us. And then, on top of that, what do you do if the tumor is like an odd shape? Because not all tumors are going to be a circle. Sometimes, they're a little bit of an oval, or sometimes there's some necrotic interiors here. And so you do your best. You find the largest measurable perpendicular diameter to the initial diameter, and you multiply that for the lesion. And if there's more than one lesion, you essentially sum it all up, which compounds the variability. So to take a step back, this variability is it's a pretty big limitation in assessing, efficacy of certain drugs. One can imagine that if there's inconsistencies in evaluating how, one radiologist would define a patient's response, it can lead to misclassification of, patient's response to treatments, which, adds noise to the conclusion of efficacy. And so what I'm trying to get at is that these are essentially two-dimensional measurements, and we believe that there's a need to develop automated measurements for 3D volumetric assessment to improve reproducibility of meningioma response assessment. And so that was the motivation for this effort. So a fabulous programmer in the group, Mason Cleveland, has taken data sets from both MGH and publicly available data sets and trained a 6-layer unit with a joint dice cross entropy loss. And using this framework, we trained 3 separate models. A model solely based off of the MGH data, another model based off of the BRATS data, which is the external data set, and then combining them together. So this work is still pretty preliminary, but what we used as a first step was to quantify dice scores to estimate the performance of each model. And so really what I want to call your attention to here is that the columns here is the model. So the y-axis is dice score. The columns here are the MGH only. This is the combined model, and this is the BRATS only model. And so what we see here is that pretty clearly models that were trained on MGH data generalize to the BRATS data set, the external data set, but not vice versa. What we also see here is as marked in the blue, which corresponds to high-grade tumors. Segmentation of high-grade tumors didn't do as well as for low-grade tumors. And in clinical practice, this may be because high-grade tumors have more heterogeneous contrast enhancement, diffuse tumor boundaries, as well as a lot of edema. So you don't have as clearly demarcated tumor boundaries. But nonetheless, we were, I think what we observed from this is that there's clearly a lot of promise in developing segmentation models for these tumors. And so our future directions are, we do have additional high-grade meningiomas that we plan on further fine-tuning the model, as well as, to identify which use cases do better or poorly. We'll do subgroup analysis, such as, where the tumor is, or if there's a correlation of like pre or post like surgery or radiation to these areas. And prospectively validate this on other data sets. And again, one of the cool things about working in the hospital setting is, hopefully we can deploy this model in the context of a clinical trial and then assess how this does head-to-head versus the 2D RANO measurements, which is the current gold standard. I don't really have the time to discuss this today, but I did want to mention it. Happy to come back at a future time and discuss this. We do have efforts in training classification models using standard of care imaging, such as brain MRIs, mammograms, to predict therapeutically relevant biology. And so, the premise of this is that the imaging signal is a macroscopic representation of tumor biology, tumor phenotype. And so we have efforts in multiple different tumor types in brain tumors, breast cancer, prostate cancers, in which we leverage imaging of these tumors with deep learning to recognize therapeutically relevant biology. What I did want to spend most of today discussing was work that uh, a very good graduate student, Tiago, spearheaded during his time in the lab, which I've called pathogenomics. So essentially using pathology to predict therapeutically relevant biology. And so to kind of give you the context for this effort, so breast cancer, unlike say brain tumors, are different in the sense that oncogenes or oncoproteins that are targetable so oncogenes and oncoproteins central to tumor growth have been identified, and therapeutics against these genes or proteins have been developed. And so one example here on the left is HER2, as demonstrated here, and that leads to cancer growth. But it's through a number of anti-HER2 antibodies like pertuzumab, trastuzumab, neratinib, we can essentially block cancer growth. Another example here is the estrogen receptor signaling pathway. This is something that we can target with a number of estrogen inhibitors to exert anti-tumor activity. And so what I'd like to highlight here is, this is the concept of individualized precision medicine. So essentially that germane biology from one person's tumor can be used to inform that patient's best choice of treatment, which may be different from somebody else's. But in clinical practice, there are a lot of current biomarkers such as HER2 overexpression, the presence of estrogen or progesterone receptor. While these inform the choice of treatment, these tools don't really predict response to specific therapies, meaning that people with the same biomarker have wide variability in treatment outcomes. And so that was the clinical motivation for this. But in recent years, we've, wet lab genomics experiments have essentially identified that RNA sequencing, or more specifically tumor and immune activity, is strongly linked with therapeutic efficacy and risk of metastasis in breast cancer for a number of treatment types. The problem is, in clinical practice, it's hard to get that RNA sequencing data because it requires fresh tissue, which is not usually able to be done because it requires a large infrastructure to do. And so, this is a problem in clinical deployment. And so we essentially sought to get that information from a widely available modality in a hematoxin eosin slide. And so the hope is that these could be integrated in clinical decision-making pipelines to maybe understand certain pathways that might modulate response and also serve as a complementary biomarker to guide decision-making. And so I guess let me take a step back. So I realize that there are folks in the audience that may not know this. So how you get tissue. So, if a patient comes in with a concern of tumor on an imaging, they'll get surgery. And then once that lesion is excised, it gets sent to pathology. And what happens is the pathologist, generally speaking, embed it in formalin-fixed paraffin so that it doesn't auto-lyse over time. And then the base stain is hematoxylin and eosin, which is demonstrated here. And the purpose of this is that it essentially allows us to visualize morphologically different cell types. So for example, here we have, I hope you guys can see my mouse. This is tumor cells here. These little red stringy things are stroma, and these little purple dots are lymphocytes. But how does a pathologist aggregate this data? So obviously it's manual assessment, and it's heavily based on essentially the tumor cell and not so much the microenvironment. So when a pathologist looks at this, they look at tumor invasion, anaplasia, necrosis, mitosis, and then really just qualitatively looks at lymphocytes and grade it as a 1, 2, or 3. And so clearly, you can see that there's a lot of inter-rater variability. And so what I am hoping many of you take away from this is that, pathologists, I mean this with no disrespect to pathologists, because my wife is actually a pathologist. Pathologists are leaving a lot of meat on the bone when it comes to analyzing a pathology slide, because they mainly look at the tumor cell and not so much the microenvironment. And so I wanted to take a moment to explain why the tumor microenvironment is important. So tumor behavior and its responsiveness to treatment is really dictated by not just the tumor cell, but also other cells that are involved or present in its surroundings, such as tumor infiltrating lymphocytes. That's what's meant by TILs here, macrophages or endothelial cells. And so this is a study that was sort of a big deal when it came out in 2016 for breast cancer. It basically showed, here are 3 different H&E slides. In retrospect, I'm realizing that this may be difficult for people to see, so I apologize. But basically in here, you don't see very many lymphocytes. Here, there's like an intermediate amount. And here, I think many people can see there's a lot of purple dots and lymphocytes. And in short, people with a high number of lymphocytes do better than folks with low numbers of lymphocytes. And this is true for a broad range of therapeutics, radiation, and a number of systemic therapies. But it doesn't just stop there. So the spatial patterns of how the lymphocytes with tumor cells, how they are arranged with each other, can dictate tumor behavior. So for example, here on the left, is a H&E slide where, we see lymphocytes and tumor cells equally interspersed throughout here. And then on the middle here, we see a bunch of lymphocytes all around here, but it's kind of, there are no lymphocytes in the middle here. This is all tumor right here. And then in here, there are virtually no lymphocytes and a bunch of cancer cells here. And so what I'm trying to convey here, the point of this is that even though the actual numbers of the lymphocytes may be similar across different slides, these spatial patterns matter in dictating tumor behavior and therapeutic efficacy. And so we hypothesized that perhaps deep learning could identify these therapeutically relevant spatial patterns on H&E that humans aren't really able to do on a reproducible basis, that can maybe lead to a way to estimating tumor and immune activity. And so we from a computational perspective, working with whole slide images is computationally difficult because these images are huge. They're literally like 2 or 3 gigabytes, and downsampling these images is undesirable because this would result in a loss of, biologically important information. And so a popular strategy that our group and I believe your group and many other groups have explored is the concept of weakly supervised multiple instance learning, which, as you all know, is a type of weakly supervised learning where each instance isn't really necessarily individually labeled, And the supervisory signal really comes from the aggregate set of instances, which is commonly referred to as a bag. And so the goal of the model is really to predict the bag label. And so in practice, what we do is we crop the whole slide image into a lot of different patches. And, from those patches, feature vector embeddings are extracted. And then these patch level representations are eventually summed up to build the patient level representation. And so to do this, this was our pipeline. We used the TCGA data set, which has paired H&E images as well as bulk RNA sequencing. And so we identified, I'll show in the next slide, 10 therapeutically relevant pathways, and then quantified the gene expression using standard bioinformatic tools. And so here what we did is, this is the whole slide image. This is a little bit of pre-processing, mostly to look for, to assure data quality. And, after which we extract a bunch of patches that are about 256 by 256 millimeters. And then, we trialed a number of feature extraction strategies. One is the ImageNet ResNet 50, which is a pre-training strategy based off of ImageNet. Another one is PLIP, which is based off of a pathology foundation model. And then we trialed a number of different model architectures, essentially one using a multi-branch attention mechanism for the classification, and the other one using a single branch for classification, as well as a transformer. And the output was essentially binary classification of these pathways. So in short, what we did is the bioinformatic tool that we use to estimate activity of each of these pathways essentially outputs a number that is the activity of that sample compared to the population average. So roughly speaking, half would be positive and half would be negative. And so here we observe that the choice of model architecture, whether it's an attention mechanism-based classification or a transformer-based classification, does not really play a major difference in model performance. However, the feature extraction strategy may play a relevant role here. So here we observe that first, the pathology foundation model pre-training strategy, as marked here in red, mostly outperformed that of the ImageNet based feature extraction strategy. And furthermore, I think a lot of us were encouraged by the fact that a lot of these pathways that maybe we expect to see signal on H&E, like T-cell mediated cytotoxicity, epithelial mesenchymal transition, which is essentially metastasis, cell cycle, which is essentially a marker of mitotic activity, did reasonably well with AUCs like in the high 0.7s and in some cases 0.8s. We didn't stop there. So I think a big thing, at least if we want to think about clinical deployment, is we got to understand what the model might be learning. And so essentially what we did here is we took this was a slide here that was correctly predicted to have a high level of T-cell cytotoxicity. And we essentially took the patch with the highest attention score, essentially the patch that was felt to be important for the outcome of interest. And we were pleasantly surprised to find that there were a bunch of tumor-infiltrating lymphocytes, which makes biological sense, given that this was predicted to have a high degree of T-cell cytotoxicity. And then the patch with the lowest attention score essentially highlighted a bunch of necrotic tumor cells. And so future directions that we are pursuing is ordinal and multitask classification. So essentially we did binary classification, but to see if we can maybe tease that apart to granular detail, as well as we essentially train 10 different models right now. And so seeing if we can, combine this to one model. We're also exploring with various data augmentation strategies, as well as fusing with other clinical metadata by which to improve performance. And the last thing is, of course, external validation with slides from our hospital and other collaborators. So now I think I'm going to turn it over to Chris.
Yep. Okay. Yeah. Thanks, Albert. So I'm going to talk about something completely different. I'll just introduce myself first. So I'm a computer scientist by training. Unlike Albert, I have no medical expertise. I've worked here at MGH for a while, and for a large part of that time, I was actually quite closely involved with the way that we deploy models and use them in practice at MGH. And this project I'm going to talk about now is kind of an ongoing project that kind of draws on that experience. And basically, the big picture is I've observed that there's a big translation gap facing people that want to deploy medical imaging AI models. So can we click through? I think some text should appear. Albert, yep. So on the left, I've kind of like Mars and Venus here. On the left is Mars, medical imaging AI research. If you're involved in this field, you'll know that we tend to use things like Nifty files and NERD files and NGZ files, and we just store our model outputs. If it's a segmentation, maybe we store it in a Nifty, maybe we store it in an MPY file, a JSON file. We communicate things via the file system, and all our software is sort of based on Python and NumPy and PyTorch and TensorFlow and these kind of tools, right? But then when you want to actually deploy something into the clinical medical imaging workflow, you're in a very different environment. It's an enterprise IT environment, and all of the imaging data is in DICOM format, which is the standard format for radiology images and also increasingly pathology images and other things as well. Things are communicated and stored using DICOM, using archives, not using a file system directly. You visualize things in a PACS viewer, and all of the software is sort of based on .NET, and it's very, very different sort of technical ecosystem. And so, I've observed that this causes a big problem. When you develop your algorithm over on Mars in this analogy, you get it to work very well, you publish a nice paper, and then you want to bring it over to Venus here and deploy it, and suddenly you're in a totally different place, and all the work you've done and all the things that you use are not compatible anymore. And this is a problem, and we want to try and sort of alleviate this and smooth this transition. So when it comes to communicating the results of your AI models, and this is your Nifty files are no good here, right? You have to work in DICOM. We're going to have to sort of play by their rules largely, and take things into the clinical environment in the way that the clinical environment works. Okay, next slide. So just to take Nifty as one example, it's by no means the only example, but I think it's kind of illustrative. If you want to store a segmentation as an output of an AI model, which is a pretty common thing to do in medical imaging, right? Like you could store it as DICOM. There is a way to store segmentations as DICOM. It's a clinical standard, it's international. It has all this clinical metadata that describes what the segmentation means, and it describes the clinical context in which the segmentation was performed, which patient it's on which image it was on. It has all the spatial metadata, it has all the acquisition metadata for the original image. But it's very complicated, it's very difficult to work with, and it's very inconvenient. On the right-hand side, there's the Nifty file format, which has become so popular for good reason, right? It's very simple, it's very convenient, it's kind of efficient to read and write, but it's not useful in a clinical environment because it lacks all of this metadata that tells the doctor how to interpret this image. Which patient is it taken from? There's no standard way to store that. What does the segmentation actually mean? Right? Is there uncertainty about the segmentation, all this sort of thing? And so there are some methods to go from left to right, but there's not really a way to go from right to left, which means that if you develop your algorithm and you use Nifty and then you want to deploy it, you're kind of stuck. So, okay, next slide. Yeah, so how should we be communicating AI results about clinical imaging? Well, I think there's a few key points. The first one is that it has to be unambiguous. Like a nifty file, if it's just a bunch of numbers, what do the numbers mean? You need to be very explicit about what things mean. Next point, it should be in context. It should have all the relevant patient and clinical metadata that accompanies. When was this image, like the image that this came from, acquired, in what context, on what date, right? All of that information is extremely important if you want a traceable medical record. It needs to use standards. It needs a consistent structure across different models and developers. If each AI produces its own output format, we're going to end up in a real mess in a few years when you need to actually cross-reference things. And we need to use standard nomenclatures rather than using free text to describe what is a tumor, what is a liver. These concepts are actually a little bit ambiguous, and you need to refer to standard nomenclatures to make sure we're all talking the same language. They need to be interoperable with existing clinical systems. Clinical systems are not going to change very quickly. These are large investments that span decades. And so we need to work within those existing systems. And that means working with the PACS in the hospital, the database that stores all the medical data, medical imaging data. And lastly, they need to be traceable. We need to record what algorithm produced the result and refer back to the evidence from which that has been derived. Okay, so next slide. So there's good news. The good news is that there are DICOM format ways of storing all of these AI results that meet all of the previous criteria. And this is not work that I've done. This is work that was done before in the past. And there's mechanisms called DICOM structured reporting, which basically allows you to store classifications, observations, measurements. And there's a DICOM segmentation format. I already mentioned that allows you to store segmentations. There's also ways of storing saliency maps and overlays on top of images that can encode sort of more general AI results. And they share a data model with the original images. The original images are in DICOM format. The AI results are in DICOM format. They share a similar data model. You can communicate and store them via the same systems, via PACS, and they can be visualized with a PACS viewer, maybe if the specifics are supported, but at least that's plausible. Okay, so good news, right? This exists, problem solved. Well, not really, because DICOM is very, very complex. DICOM structured reporting is particularly very, very complex. Is just an example. This is what a structured report can look like. It's a very highly nested set of different metadata with a very complicated structure. And, as an AI developer, do not want to have to go and read and figure out how to create these things, right? You're going to probably make a mistake, and it's going to take you a very long time, right? So, okay, so next slide. And there are no tools really to do this. So that's where this is where I've been building up to. This is a project that I started along with a colleague of mine, Marcus Herman. He worked in the pathology department. I work in the radiology department. And so we decided to build this library. We call it HiDICOM. It's open source. And it's an open source Python library for creating and reading these derived DICOM objects. Derived meaning derived from images, so they encode sort of AI results. So it can deal with segmentations, structured reports, which allow you to encode classifications and measurements, parametric maps, secondary captures. And so we released this a couple of years ago. It's now getting downloaded something like 4,000 times a week or something crazy like this. So it's getting very popular. And this is not an entire solution to speeding up this deployment process, but I think it's an important part of the process. Being able to take the AI results from your model and encode them straightforwardly in DICOM format so that you can send them to a clinical system. That's really what we're trying to achieve with this library, and then also be able to read them back after you've done that. So yeah, next slide. Yeah, so the goals of the library are to make it as easy as possible for machine learning scientists like me and like many of you to work with these derived DICOM objects. We want to make it interoperable with the existing ML ecosystem. So there are a few other tools out there that do this, but they're mostly C++ libraries. And, if you're as a Python-based ML developer, you really want to be working with Python and NumPy and PyTorch and TensorFlow, right? And we want to make sure that anything that our library creates, any DICOM file that it creates is correct, which is not trivial because these things are highly there's lots of constraints upon them. There's lots of interdependencies between them. And it really doesn't shy away from making you as a developer provide all the necessary clinical information. And another important goal, because of our backgrounds, was that we wanted it to treat radiology and digital pathology both as first-class citizens, not focus on one over the other, but realize that to the extent that we can bridge these two fields with similar standards, it's going to be very helpful for the field, especially some of the work that Albert was talking about earlier. So this was from a paper that we published a couple of years ago when we introduced the library. We're still, it's still under active development, but I think this is still a nice experiment that we did at that time, where we basically trained and validated two different models for lung cancer assessment. One was based on CTs, where we trained a lung nodule detection algorithm. I mean, nothing fancy about the model, but and then on the right-hand side, we trained another system to detect tumor regions and classify the histology of lung cancer slides. But the point is that we did all of this using reading from and writing to DICOM format annotations and results the whole way through. So the whole way through this experiment, we were essentially compatible with clinical systems, which is very different from the normal workflow that a lot of AI developers are used to working in. So we showed that it's possible. And as I'll leave it there, but the library is continuing to be developed, and we're working closely at the moment with the Imaging Data Commons in the US, the NIH's Imaging Data Commons, which is standardizing a lot of cancer imaging data in DICOM format. We're working very closely with them and making the tool usable for them. Okay. I was going to squeeze in this at the end, but I think we're running out of time, so maybe let's leave this out and end it there.
I too will be brief. We have a few last slides. Chris and I have a number of collaborations with other people in other institutions in America, but also around the world. And, in short, we think there's a lot of opportunity for both clinical and technical innovation. We do have efforts. I think this also opens up opportunities for efforts in federated learning, and which we've explored in prostate cancer. Another thing that I'm going to be quick here for time restraints. Another interest that we have that we didn't really have time to get into is using machine learning as a tool by which to integrate, high dimensional data. And so for here, this is a project that a very talented master's student in the group, Tobias Bondenman, is essentially trying to predict therapeutic response of a specific drug by fusing together radiology, pathology, and clinical metadata information. So when you think about it, each modality contains different biological information. And that is under progress. And so this is our last slide. This is kind of, our dream for the future. So, I think the current standard of care is what is the right way, or what is the current standard of care way to integrate multiple modalities of information? Well, it's through a tumor board, because humans are clearly very adept at integrating this information. But there are challenges as listed here. And so our hope is that, can machine learning be used to augment human performance for improved decision-making? In my opinion, like integrative multimodal fusion is a little bit underdeveloped because there's a lot of biomarkers right now are kind of reliant on one modality, but that's something that we're keen to explore and collaborate with others. And so with that, we'll leave it there. I'm just a physician. And so like a lot of the work that we do is multidisciplinary. And so we have to thank, the rest of our team and collaborators for these efforts, and happy to take any questions.